Good morning, everyone. Good morning, YouTube. As soon as I get the thumbs up from Megan, we will get started. Make sure our stream is actually streaming. We are good on YouTube. Okay, so we'll get started. It is getting to be the end of the semester here. It's our next water, wetlands, and watershed seminar. So today we are lucky enough to have with us Dr. Zach Siders. So Zach is an assistant professor in fisheries and aquatic sciences here at UF, and his research looks at diverse quantitative approaches. So are you gonna show us the math, or are you gonna just?
Oh,
Okay, so
there's one equation.
there's a lot of quantitative ecology, especially over the last 15 years or so. We should be okay there. And so we use these quantitative methods to get robust ecological inference, and hopefully that means that if we understand the system better, we got to know the math so that we can do the ecology, so we can manage the ecosystems better. Much of Zach's work centers around fisheries ecology and management in the Pacific and Southeast US. The life history of elasmobranchs.
Yep.
So raise your hand if you know what an elasmobranch is. Are we gonna know now?
Sharks and rays.
Sharks and rays, there you go. Finfish and turtles, and the aquatic biodiversity of freshwater rivers and oceans. He has his PhD here from UF in 2017 and his master's from UNC. This is University
Wilmington.
in Wilmington,
Yep.
yeah. And his BS in biology from UNCW in 2011. So Zach, the floor is yours. Thank you.
I think you got a couple folks in here.
Oh yeah, all right. We better admit them. Okay, so there we are.
Thanks for coming. Thanks for those online. Call me Zach. I'm a quantitative fisheries professor in fisheries and aquatic sciences. As David mentioned, I'm mostly do a lot of fisheries ecology today, but or on my day-to-day, but we have been working with some really great folks at the Turtle Survival Alliance, which is an NGO. They've been running long-term mark-recapture studies in a lot of sites across the Southeast. And I was fortunate enough as meeting them through some sea turtle work and then eventually getting involved in a bunch of freshwater stuff. So we'll talk about some work we've been up to recently. So first off, what is the role of turtles in the ecosystem? So turtles are interesting in that they're generally very long lived. We have things that might, like this little musk turtle down. This little musk turtle down here in the lower left. They might live like 5, 7, maybe 10 years to something like the snapping turtle in the top right. They're living, 50, 60, 70 years plus. We have some tortoises, 300 years or so. So they can live for really long periods of time. They're pretty abundant in most of our freshwater systems, which is kind of interesting.
Gotcha. Here we're good. Okay.
For such a long-lived species, they generally are living longer than kind of the other really abundant predator within these systems, which would be fish. We have really varied diets. We have things like in the top left and the bottom right, we have these herbivorous species. They're mostly eating aquatic macrophytes and algae and periphyton. And then we have kind of on the cross our kind of really carnivorous species. We have kind of a piscivore, scavenger, and our snapping turtles, and our little molluscanivore and our little musk turtle down here in the lower left. And so as these kind of abundant, long-lived species, they can really integrate across a lot of our kind of indicators of ecosystem health. At like a year's scale, they're producing hatchlings. And so that kind of indicators of how many nests are they putting out, where are those nests happening, what's that nest success rate, how fast are those hatchlings growing can tell us something about what's happening that year. Versus over the lifespan, we can start looking at things like how fast are these species growing, what sizes are they getting to, what's their kind of diet and health composition, what's the rate of diseases, how often do they reproduce, and those things can tell us things about the ecosystem over longer scales. The system we're talking about is Comal Springs. We're going to touch on how this is a little bit related to our Florida Springs near and dear to our hearts here, but Comal Springs is in Comal County, which is, if you look on the left-hand map here, this is the geology of Texas, and you can see basically there's this like green and then a kind of light green and an orange all lined up compressed there on this fault zone. It's the Balcones fault zone. And this Comal Springs is part of the Edwards Aquifer, which has a huge area that is contributing in the gray in the middle map here. And all that contributing zone, as well as this kind of unconfined zone, acts a lot like our aquifer here in the Florida aquifer, where we have kind of rainfall off of a really large scale, contributes down, percolates into that aquifer, and then creates underground flow. And that underground flow pops up in a few, very few springs compared to our Florida springs here, where they're popping up in just a few of artesian springs, including the largest one, which is Comal Springs. It's the largest spring in Texas. It's the largest spring in the southeast. And it's located, unlike a lot of our springs, literally in the smack dab middle of New Braunfels in Comal County in Texas. And you can see that with our left map here. That little red area is where Comal Springs Land Lake is. You can see all the development. There's like a highway passing through that goes down to San Antonio. This spring is literally in the middle of town. There's like residential areas around it. It is really, really developed around there. And like most of our springs, they also have a lot of spring heads that all contribute to kind of what we're calling the spring. In this case, there's basically 3 large, 4 largish, 7 medium, and 1 small springs. Most of those are up in this this area in the middle here. See pointer, there you go, up in this area here. And together they basically form, and this is pretty common to Texas springs, these big lakes. So it's basically, you'll see things like Spring Lake when you're looking across some of the Texas water features. And this is no different. This one's named after a guy, John Landa. We'll talk about here in a sec. But you have this upper spring run that starts over on the kind of right-hand side of this middle graph. It flows and then forms the lake, and then it outflows into the Comal River, and then that Comal River flows to the Guadalupe River, and then that flows into the Gulf. So that's where we're at. It is a first magnitude spring, and it would be right behind Itchituckney, which if we were to include it in the Florida list, we'll put it in our kind of Florida top 10 springs. It is highly variable in output, more so than what we tend to see in our Florida springs, including this historic drought that you see on the right-hand graph, where in the 50s, it basically went almost completely dry. Basically, only the spring heads and like a really tiny area was actually had water in it. And we've had some droughts over time. And one thing to note is those droughts seem to be happening a little bit more frequently than maybe what we had seen them in the past. We're in a one right now in Comal at the moment. It's almost as bad as the 1950s one. It has been inhabited by humans for a really long period of time. The Qualhitican Tayaya people had a main settlement here. Their main settlement, their big settlement was near San Antonio. And in 1691, when the Spanish made contact as the first kind of European contact with these folks, they found all these people who were using this place as a Comal, a cooking area. And so this is kind of what's depicted in our painting here. That's why it's named after it, and the county's named after it. And this is a hunter-gatherer society. That's kind of cool. They specialized on pecans, and this is a really rich pecan area. So they were harvesting pecans, and they were using pecans for a whole lot of their food and kind of consistent subsidization. And there's a food processing site, basically the one that's depicted in this painting, that you can still go there today and see like residue from them using this place for presumably hundreds or not thousands of years. So of course, the Spanish being what the Spanish were at that time, they built a missionary. Then they like the land so much that they give it to the governor. This is kind of the classic pattern for Spanish colonization. Then, of course, some other European settlers get involved. We have some German folks who move out there. They end up buying land from the Mexican government at that time. And being European settlers, they do what European settlers do, which is to blow the springs up to make them flow more. They also use slave labor to dig a bunch of mill races. And this is an example of what that meant. So this is from 1879, a little like drawing pictograph of what it is. And that's a lot of water coming out of it in a giant mill race. Like that would be impressive to see if we got that out of one of our Florida springs. They had the topography there, unlike us. At the time in 1860, there are 7 grist mills, flour mills, sawmills. There's an ice like factory. There's a brewery. There's all this stuff happening in this property. John Landa, which Landa Lake is named after, he buys it in the 1860s, and by like the late 1800s, it's already known nationally as a giant recreational area. So it's like, go to Landa Lake, that's the cool place. The city opens Comal Bass for public swimming. That's in our little sepia tone photo up there. There's like steamboats operating on this thing. It's like a whole on like amusement park happening at this place. In 1920s, they get a record 800,000 visitors, which is a lot for 1920. Land and Park is sold in the 30s, late 20s, and also a giant power plant shown in the lower left is built there. The Depression causes the park to close. So then in the mid 30s, the city buys the park and they've owned it since, and they operate it with the Edwards Aquifer Authority. That historic drought we were talking about earlier reduces that spring system to such little isolated pools that the endemic fountain darter is kicked out and extirpated, and then later on, once the spring comes back, they reintroduce it from other areas. That's our setting, but what the hell is a gilded cage? So the gilded cage is inspired, the phrase the gilded cage is inspired by this oil on canvas painting by Evelyn de Morgan. And it depicts this woman living in a life of luxury, fancy gowns, nice manse, the whole nine. And she's staring out this window of a group of dancers, a little bit more scantily clad than she is. And they're led by this woman, and there's this nice soaring bird over them versus her little gilded cage songbird in here. And so that's where the gilded cage phrase comes from. And it's a luxurious environment that fails to provide all one's needs. So what we're gonna do is we're gonna walk through the gilding of Comal Springs, what makes it nice, and then we'll talk about what is the cage of Comal Springs as we go a little later. So the gilding. Like our Florida Springs, Comal Springs is just chock full of submersed aquatic vegetation. It's a polyculture of algae, mostly turf algae and periphyton. Has a lot of macrophytes. A lot of like our Florida Springs, it's dominated by Vallisneria. It has also these really cool bryophyte mats that form in the upper spring run that look like little like fluffy little Brillo pads. Unlike many of our spring runs, it still has this really, really intense SAV cover. A lot of our springs, you go to like Manatee Springs, it used to kind of look like this top photo here, and it no longer does. It looks like the same bottom. This system still has it. So it's kind of an echo of what Florida Springs used to be. This cover varies throughout the system. Some of the other macrophytes we have are like Combamba, Luigia, there's some lily pads and Nufar around, but the dominant in that light green and that kind of cover map is Vallisneria. And so if you have a bunch of Val and you have a bunch of submersed aquatic vegetation, you have a bunch of turtles that like to eat it. And one of the groups of those turtle are Cooters, the Pseudomase taxa. And what we looked at was we compared our Pseudomase taxana, the Texas River Cooter in Comal Springs, Land O'Lake. We compared that to 4 other systems, all of them being Florida. We have Fanning Springs over on the Swanee River, Manatee as well, Wekiwa Springs, which is over on the Wekiwa River, and Blue Springs. And we looked at 4 different taxa that kind of all inhabited at least 2, with the exception of Texana here of those spring systems. The Turtle Survival Alliance, not me, caught a lot of turtles here, which makes it really nice as a quantitative person, because now you have lots of really cool data. Those are the number of turtles that we caught of each of the 2 sexes and the different years that we captured them. This was a few years ago. I think we cut it off in 2020 was the data that went into this paper. And so Wekiwa is interesting in that we just hit 25 years last year of monitoring this system. And it's one of the longest mark-recapture data sets probably out there.
Are you gonna say what mark-recapture is?
No, I can.
Yeah,
sure.
Do you guys already know?
You know, Mark, okay, so we'll talk a little bit. So what we do is they go out, and we'll talk a little bit more about this, but they go off, they survey, they grab a turtle. You take that turtle, you can do shell markings by doing like a file or a Dremel on them, and there's a certain pattern to labeling their scoots along the back. And then they also put a pit tag in that you can scan. That's like what you get for your cat or dog at the vet. And so we have electronic tagging them, they weigh them, they measure them, and then we release them. Typically, we also then get them the very next day when they do like 3 days in a row of these surveys, because the turtles are hanging out there. So you get a lot of interaction with these turtles over the years, and you can get some really great data sets. One of the turtles in Wekiwa, I think, has been caught for at least 19 of the 20 years.
So they capture it, they can mark it, and then if they come back 5 years later, if it's the same turtle, they can see it.
Yeah, and we can look at how they change. So how are they changing their growth? So we look at every individual and we fit individual growth models to them. They all have this kind of asymptotic form. So they ascend pretty rapidly and then they flatten out on this graph here. And so what this tells us is that we basically have turtles who are growing pretty rapidly, then they hit maturity and they start taking all that growth and basically how big they are, and they start investing it into reproduction. And so that's why the growth slows here on these graphs. So we look at every single individual turtle. We can build basically a history of that turtle's growth over all the times, 19 years in that one individual's instance. We have, I think for that individual, I think it's close to like 46 growths that we've actually were able to measure. So we capture multiple times a year. And then what we can do is we can fit these models to all the individuals and get estimates for males and females for both of the 4 different taxa that we're interested in. The main takeaway here is this purple one down in the lower left. This is our Texas River Cooters in the Comal Springs Land O'Lakes system. And what you can tell is the L infinity is where this line asymptotes out, where it flattens out on this graph on the right. So where that line flattens out, that's your average maximum size. That average maximum size is way bigger than what we see in non-spring sites. So we're getting like 313 millimeters maximum size in a spring versus 255 out of a spring. And so we're looking at like 120, 130% increase of just
like in some other river.
In some other river. This is nearby in a river in Texas. And then if we look at, that's for males and females, it's like 120, 140% increase of just how big you get in a spring. They also get there way, way faster. So not only do they get bigger, but they grow much quicker. And we can see that if we just look at how much growth they put on in the first year. These are insane growth rates. Kind of the average for a turtle of first year growth will be about 20 to 30 millimeters maximum. In springs, we're seeing turtles put on like 60 millimeters in the first year. So they're hatched 35 millimeters or so a little bit like an inch and a half. And they're getting all the way up. They're adding almost like 4 inches over that time, right? So they're getting huge in that very first year. And it's all due to that really intense ability to prey and resources in those springs for them to get provision. This is nuts in that this turtle's growing. Texon in this case is growing 18 to 20% of how big it will ever get in that first year of life. And this turtle's living 30 years. 20% of its growth it's laying down in that first year at a spring, which is crazy. This is faster than humans who are being provisioned by their mom and also don't have to grow this lovely shell. So it's quite a bit of growth. I don't even know. I mean, it helps that there's, it's a pretty calcium rich environment in a spring hen. So that probably helps them grow these shells quickly. We can look at another species. This is Stenotherus odoratus. It's the same procedure, mark-recapture data sets. In this case, this is a whole bunch of folks. We're all coming together to work on Stenotherus growth across their entire range. They range from Florida in the east all the way up to New Brunswick in Canada. They go as far west as Texas, including our red, which is our Comal Springs site, and they go all the way up through the Great Lakes. This is really cool in that we have basically a mammalian Bergmann's rule. The Bergmann's rule just says mammals get bigger in the north and they're smaller in the south. This applies to mammals. That's where it was built for. We don't have a turtle Bergmann rule for variant of it, but they basically are bigger in the north and smaller in the south in most of the cases, with the exception of Comal. And Comal is so much bigger than even a spring site next door to it, which is in Spring Lake, and that's this little one down the lower right. They're getting 109 millimeters in their maximum size for these musk turtles versus 95 millimeters. So they're going 14 millimeters larger in just Comal versus another spring, basically next door. And they're growing almost an entire inch bigger than what we see in our Wekiwa Springs in our yard. So Comal is great for turtles. That's what the takeaway here is. Comal makes big turtles, it makes them fast, and we get to see a whole lot of them. Why do we see them? Well, we have that submersed aquatic vegetation, but our little musk turtle here, he doesn't eat veg. What he does eat is he eats snails who eat veg. So that the really robust vegetation is grazed upon, including a lot of the algae that lives off of it, on, in this case, native snails and an invasive snail that got introduced in the 70s. And the turtles here, these little musk turtles, they grow megacephaly. That means their head gets huge. So if you've ever been to a spring here like Itchitutney, and you've seen little musk turtles swim around. They're usually light gray and they have little black dots on them. That's the loggerhead musk turtle, Stenopterus minor, closely related to this guy. These turtles look like the giant Stenopterus minor heads that we get here, which is weird because they all usually look like the little guy on the right with little tiny heads. And they've evolved this so that they can eat this slightly larger invasive snail, which is kind of crazy. And they did this since the 70s, so about 50 years.
There's a local adaptation.
Local adaptation to an invasive species, and very similar to what we see with snail kites or apple snails here. So we're seeing it here. They've sampled around Comal to look for this snail and other Snothorus. That's where this photo comes from. This is a Snothorus from nearby, literally just outside the system, and he's a regular looking one. So, and you're gonna see the size difference. These turtles are probably about the same age. And just how much bigger the one who is getting a bigger head and a bigger body as a result of this invasive species subsidization. We got a lot of turtles growing fast, getting big. We also get a lot of them. So this is an example of one sample of just our musk turtles there. You can see there's some tiny guys in that kiddie pool bin thing that we got going on here, as well as some really big turtles. That's about probably the maximum size that you would ever see of these odorata. And as a result of basically over the 10 years that we're doing this mark-recapture study in Comal Springs, we'll talk a little bit about what that looks like here in just a sec. We see in total over those 10 years, our model estimates, we'll talk about the model here in a sec, 44 common snapping turtles, 1,500 or so Texas river cooters, 6,000 or so musk turtles, and 504 red ear sliders. This is not a big place.
This is the population estimate.
This is like how many turtles were there during that 10-year period. That's a ton of turtles. I mean, this is like 6,000 of these things. It's like the bottom is mostly turtle, not dropfish. So it's a lot of turtles, super dense. So we wanted to kind of get an idea of what was happening at Comal, and we entered into this kind of in like a little bit of a celebratory pattern. Like, hey, we've done 10 years at Comal. We did a 5-year assessment. Let's do a 10-year assessment. This place is gangbusters. The turtles are doing awesome. We got these weird musk turtles with their giant heads. Like, let's go in and kind of take a look to see how Comal's been. We were not going in being like, this place is bad. That was not the idea. And so again, that North American Freshwater Turtle Research Group with the Turtle Survival Alliance, they did all the sampling. They sampled a ton, sampled 96 days over this 10-year period. 34 individual capture events. That's a 3-day block of sampling together. They have 2, that's over 10 years, and on average, we're about 110 days apart. We have a couple that are a little bit less than 2 months apart, and then we have COVID, which gives us our one that was over a year. In that time, they interacted and ID'd 4,477 individuals. They had touched 8,740 captures in that time. 6,043 or 6,435 recaptures, and in the end, we recaptured 2,000 individuals. So we were able to get histories on 2,000 individuals. So like, I went out on this day and I saw you, and then I went out on another day and I saw you a little bit later. That's that mark recap pattern. So if we just look at when we get a new individual. So we're looking at, we get a new adult, we get the turtle, we go out in the survey, we grab the turtle, we can sex the turtle, we can do that with secondary sexual characteristics. That means like their tail's a little bit fatter or a little bit longer, we can tell the different sexes apart. This is kind of an inexact science, it's kind of a field measurement that we can get. We're not like pulling blood to do sex later or doing histology, this is just in the field. So it's kind of they tend to move around just a little bit, especially when they're small. This is only sexed individuals. So individuals we can catch, we know they're an adult because we can sex them, they've matured. And what we see is we see really different patterns on when we catch them. On the top is our snapping turtles and kind of the blue teal. We see that we kind of just get a new turtle. It's like 1 to maybe 3 turtles every single time that we go out. Each one of these bars an individual capture event, one of those 34. You can see that we just kind of get turtles trickling in over time. This is because snapper's a little bit hard to grab with a snorkel survey. Snap. So not exactly the ideal way to go about it. If we did a hoop net, we could probably have gotten all these turtles. So you basically go and put a net in, bait it. You could have gotten all these snappers in one day. For our Texas river cooters in the yellow orange, and then our musk turtles in the green, what we see is we basically have a bunch of turtles in the start. That makes sense. There's turtles in the system. The first time we go out, we get a bunch of new adults. And then every time that we go out after that, you basically see that drop off. Now that's expected. We should see a kind of drop off and then level. What we don't want to see is what we see kind of at the tail end of all of these, where it kind of drops off and goes to zero. We would expect that we would consistently get new adults as they grow or they immigrate into the system, but we don't. We basically, once we get to COVID and a little bit after, we're basically getting no new turtles, no new adults coming into our surveys. Our red-eared sliders, they're a little different. We kind of have them variable over time, but what you can tell is you see that we go from some high captures early in that period on the purple red to kind of some lower captures over time. So we're still getting them. We're kind of leveled off, kind of what we would expect to see. So this is concerning just on its out for our river cooters and our musk turtles. We're just not getting new adults. And you're like, why? Where are they coming? Why are they here? What we do to map those dynamics over time, so those dynamics of those different turtles, is we use this multi-state Jolly Seaver model. So we'll walk through this just a little bit. I'll jump over the map so we follow along. This model is one of a standard series set of mark-recapture models that it can use. The advantage of the Jolly Seaver here is it can deal with open populations. That means that we don't have to assume that Land O'Lake is closed. No one can come in or out. We can assume that new individuals move into this population through either growing up, hatching and reproducing and then growing in that system, or they can immigrate into that system. And we can call those turtles that might enter into the system, they're in this not yet entered state. So they're out there somewhere floating in the landscape, or they've hatched and we haven't quite been able to sex them yet, and they're swimming around in that system. And then once we're able to grab them for the very first time, we can move them over into this blue state, which is alive. And in the case of this model, we separated that blue into two other little states for males and females. So we have alive males and alive females, because to get into the alive pool, we had to catch you, mark you, and sex you. Then basically what we have is turtles the only way out is to die or leave. And in this model, in this case, we can't tell the difference, because we don't have data on it, between whether you died or you just were like, I'm done, this Comal thing's not working out for me, I'm leaving. We'll talk about why we think that's probably mostly death a little bit later. For all those states, that's what the model's trying to estimate from all the data that we have of these basically, I saw you, I didn't see you, I saw you, I didn't see you, these series, these capture series as we call them. And those come from this observation process of I went and swam my snorkel survey and I saw you, I got the individual, we scanned it or we read the marks on it and we said, yep, hey, I got ID 23. We got them again. That's our scene in the orange. And if we don't see you, we get a 0. Now, the cool thing about these models is they're restricted. And what we mean by that is there's only certain ways that you can get a seen turtle, right? If you haven't entered, we can't see you. And those solid arrows from the green to the open orange circle say, we know we can't see you. You have to get a 0, right? It's deterministic. If you are alive, then we have a stochastic process of detection. Did I catch you or did I not catch you in that time? And that's those dashed arrows there from the blue to the orange. And then again, we have a deterministic process. If you died or you left, I can't see you when I do a survey. And so the way that we represent this is the green area, the green states, we call the superpopulation. Those are kind of the numbers of the total number of turtles that we were talking about earlier. You can think of this as basically a waiting room. These are turtles that are either going to grow up or immigrate into the system, but we haven't gotten them yet. Then once we get them, they leave the superpopulation and become a male or a female via this gamma parameter. That's the one that looks like a Y here. And that is an entry probability. So they're entering the population. And then they have to survive, and that's that phi, the circle with a line through it. That's that phi parameter there, and that allows individuals to survive from one capture to the next. Now, a lot of these models are originally designed for basically one survey a year, ideally in the exact same time of that year. And so you go out every spring, every year, and that's how you would do it. And so you'd have 1 point per year. And what you wouldn't have to deal with is time, because you don't have any raggedness. No, the time points aren't at any different points from one another. But as we already saw, We have ones that are 45 days apart or 49 days apart. We have some that are 400 days apart. We want to be able to bring all that information. So what we can do here is we can basically raise that little survival parameter to the fraction of the year between capture events. And that allows us to deal with this ragged time. Super nice way of handling this. And then again, we have to be detected. That's our little P parameter down here. And that moves us for every capture. So I promised one equation. This is what all that math looks like in a equation here. These are state transition matrix here. And so what we can see is basically if you haven't entered yet, you can only enter into the alive state with that gamma parameter, that little y there. And then if you're alive, the way that you stay alive is by surviving. The way that you die is 1 minus surviving. So the opposite. And then if we're over here and we have our states of not yet entered, alive, and dead, we can only, again, we can't see you if you're not entered, we can't see you if you're dead, and we can only see you if you're alive. And if we don't see you, it's 1 minus our chance of seeing. So that's the way these models work. One of the challenges for us is that we have snorkelers, and not all snorkelers are created equal. Right, and not every survey is created equal because we don't have the same number of snorkelers or the same people who are doing it. This day is cloudy, our visibility is different, whatever. There's lots of things that impact that snorkel survey. It would be a fool's errand to try to model our way out of that, to try to build a model of how all that stuff varied, because you could have some weird relationships where it's like if we have too many inexperienced snorkelers all in the spring head. If you snorkel a spring here, you know exactly what I'm talking about. They kick the bottom up, you can't see anything, and then all of a sudden we can't see any Sinopterus odoralis, right? So how do we model our way through that? Do we like give them like a skill test where they have to like find a turtle that we made up on the bottom? Like I don't know how we work our way through that. So, and it would be impossible with volunteers. It's like, oh, before you go snorkeling here, take our skill gun over here. No, we're not playing with you. So to deal with that fact that they're inherently noisy, what we did was we did this time correlated random walk. All that really means is that every single capture event can basically have a random chance of detecting individuals. And those that there's a distribution that controls how variable that detection is at any given capture point. And so if you look at this graph here on the right, you can see that every single capture point's bouncing around, particularly on those red and purple lines there. That's for red-eared sliders. And so what this allows is the model allows us to kind of track those changes and how good snorkelers are and how many we have and what that variable kind of effort is that's being done on every survey. The time correlated part just says, hey, if last time it was like 35% detection, the next time is probably going to be somewhat close to 35% because you're going to have generally the same pool of people, probably the same weather conditions, those sort of things. And so it just basically says there's some sharing of information between them. And that's what's really, really useful about this technique. The cool thing that we did for this model that's different than most of the models is we ran it with all species together. Usually you run one species at a time. We pulled all the species together and let all the species inform basically how variable the snorkelers were every single time. And so the different line kind of where they cross that y-axis, that y-intercept there, that's set basically globally for the species. So each species can have a different kind of raw interaction rate when we go out there, but how it changes over time is shared with species. And that's because we have a few species where we get very few like snappers. We're currently doing this for manatee and fanning right now for some dead cells. So that kind of handles how good we are at seeing turtles. We saw kind of a general decline there, kind of hop back, kind of goes down over time. That probably indicates that turtles are getting shy, called trap shyness. So we've gone out there and grabbed them so many times, like, yeah, maybe I don't want to be around that snorkeler guy. I got picked up, he threw me in a canoe. I had to wait around for a while, then he poked me and threw me back. Like, not the best thing in the world that I want to deal with. So that might indicate some trap shyness. One of the indicators of that is if you look at this dip, we're kind of at the lowest period here in 2019, end of 2019. We then get COVID, and all of a sudden that bumps up because we're not in the system for a while. So all the turtles like, hey, I haven't been harassed by that snorkeler guy for a while. That's great. And then we get a little bit naive again. That's what we call it. So once we've handled that, we can look at the entry probabilities and we can see that what we see is, again, like our kind of raw number of adults coming in, we get a bunch of turtles. Those lines are all really high in 2012 on the left-hand side of all these plots. So we have lots of turtles who are already there when we get into the system. And then we basically have those lines kind of swiggle across for a while for most of our species, with the exception of the blue snappers here. And what the main thing to point out is at the end for our Odoratus, our musk turtles, and our Texana, those lines are basically flat, which indicates that our entry probabilities are really low. They're pretty consistently low, which is bad. So it basically means we're not getting new adults. So we saw that in the raw. The model's picking up that same pattern here. So we're doing a little check here on our model that says, hey, I saw that over in my like kind of raw ticks. We're seeing the exact same here. We do again see a bump for almost all the species for post-COVID, where we're getting a bunch of new adults who kind of come into the system and are probably naive to being snorkeled and trapped. But we basically stopped getting new individuals of the Texas River Cooters and the Musk Turtles by 2017. And again, that's adults. When we look at those adult populations over time, each one of these, this is population size. So the output of those models is an estimate of population size that we get. We see that we're increasing for our blue common snappers here. What that is just us eventually finding all of them. So basically by the time we get to like 2019, we basically have found all the snappers that hang out in the system by snorkel survey. And then it flattens out after that. And we basically say, okay, well, that's the number of snappers, and they're not really changing over time. Again, these snappers live probably 40 to 50 years. So it would be rare for us to see a death of an individual within a 10-year period. That would be pretty bad, at least adults. For the other ones that we get are our orange Texas River Cooters and our green Odoratus musk turtles. We get basically a rapid increase as we're discovering individuals in that population. So we basically discover individuals, that comes up, then it kind of flattens out, and then it declines pretty rapidly in our Texana here, our yellow River Cooters here, and basically declines really rapidly for the little bit darker orange females. Our musk turtles hang on a little bit longer. The males start kind of declining around 2015, but slowly, and then it kind of picks up speed over time. The females hang on to like 2018, but they're already at a much lower number than the males. We'll come back to that here in a sec. And then they decline pretty rapidly after 2018. We see the red-eared sliders. It takes a little bit longer for us to kind of discover them. They're a little bit more mobile. I think they're moving in and out of the system a little bit better than the other turtles. That tends to be the case for red-eared sliders. It's one of the reasons why they're invasive across a lot of areas. But then also they decline kind of in 2020. And this is kind of interesting because if we flip back one slide, we see that we get this pulse. The model saying, hey, there's a whole bunch of new red-eared sliders coming in. That entry probability bumps up. It says, hey, there's a whole bunch coming in. And then we come over here, you're like, but it's still going down, right? And what that says is basically we've had new individuals moving in, but we're losing all the individuals that were there. So we basically have almost turnover of who's in the system at this time, which is also kind of concerning. Basically, whoever was there died or left, and we just get some new ones who were kind of not very wise to the gear yet. When we look at the sex ratios, the differences between those population estimates over time, we get a male biased serpentina, so their line is below that one dashed line there, which would be equal males and females. But we tended to gain females over time. Females tend to be a little bit smaller in this species. The males tend to be bigger, and so we're probably it's a factor of us finding them while we're snorkeling around. We're just eventually get the bigger males because they're easier to see, and then we eventually grab those females. We look at Texana, that was that one that really declined pretty rapidly. We actually started off with a positive female bias ratio, so we're like 6 females to 5 males. And then by the end of that time series in the orange there, we're looking at 3 females to 5 males. For our musk turtles in green, we basically were always female biased. At best, when the males were declining a little bit earlier than the females, and the females kind of hang on there, we creep up to this peak in 2018 of basically 3 females to 5 males. And then by the end of the time series, we're down to 2 females to 5 males. So we're basically losing females in both the Odoratus and the Texana here, which is interesting. We gain a few females in the red-eared sliders over time. The males, again, seem to be the ones that are moving around a lot. The last thing that we looked at here was, we've all done adults up to this point. That's all just adult stuff. But our snorkelers are able to get juveniles, and we're just not able to sex them. So what we looked at is we looked at two features. On each of these graphs, there's a bunch of box plots here, the bars, and those indicate what the range of turtles that we were catching that we weren't able to sex. So these are mostly small turtles, and it's just the range of sizes that we're able to catch. So you see when we first come into this taxona system in orange here, we're generally catching a bunch of turtles that are right about that size where they're gonna mature, and then we would be able to sex them. At this point, they haven't matured quite yet, and we're still getting them. You then get a dip in the next period, that's probably new hatchlings coming into the system, then it kind of pops up again. Then we have another nesting event, and we get the dip again, and then it rises, then it dips. So we're tracking some of those nesting patterns as turtles are nesting, hatchlings are still coming into the system, and the dots tell us how many turtles. And so we go from lots of hatchlings coming in, like a peak of 27 hatchlings coming in for these Texas river turtles, to basically none in the latter time period, like 1 to 2 maximally. And we've basically been sub 5 hatchlings moving in on a given capture event since 2017. So we're not getting new juveniles either. When we look at it for odoratus, it's a little bit more variable in green here. And we basically have 1 to 10, maybe a 20 in 1 year. And then we're basically back down to like 1 to 2 and very few tweaking. Going more capture events with none capture. And then same for our red ear sliders. Basically have 1 to 2 that we're getting. Again, these unsexed individuals, they tend to be small. And then we basically don't get any in the last period. So the takeaway here, what's happening in this system is that we're not getting new adults for basically 2 of the species. We're getting maybe turnover in our red ear sliders. So musk turtles and our Texas river cooters are declining adults. We're not getting new ones. Population sizes are going down. Our population size of red ear sliders is going down, and also probably maybe swapping individuals within the system. We see that turnover pretty intensely. And our common snapper turtles, we can't say anything because they live forever. So let's look at the cage. This, in the little blue strip in the middle of this thing, is Comal Springs Land O'Lake. It starts up here at the top of this figure in this little L-shaped thing. That's the upper spring run. It runs down, and then this is the lake system here. Right here where my cursor's at is a weir dam structure, and then it flows south from there into the Comal system. Actually, right down, I believe it's here. Yeah, I think it's down here, or one of these, no, over here. They actually have a tube chute for tubing like we do on our rivers, and it's like this giant concrete water slide that you can do. So kind of a cool thing if you're ever out there. But yeah, it's residential and other development, and then a giant golf course next to it. So not a great macro view for these turtles. There is this little respite here. There's kind of this like little riparian edge, and then the riparian corridor that forms here. This is Panther Creek to the north. You can see how narrow those systems are as you kind of move south too.
Where's the spring head?
What is this? This is in New Braunfels, Texas. So this is the Land O'Lake system here. This is
Where's like the spring discharge?
oh, the spring heads are all right along this, and then also up here as
Up
well.
there.
But they're sampling basically from this end here all the way down to this dam here. So we're not going south of that into the river proper. If we zoom in on this system, the upper spring runs on the left. What you can see is there's some old structures that were built that are retaining walls, old stone retaining walls, that this is at the upper spring end. There's like people literally walking right next to it. So pretty different looking than if you like went to Itchitopee. Not exactly like, here's the rock wall we built around the spring head. This is what Convict Springs looks like if you ever go there on the Swanee River. It's literally like looks like a swimming pool. You really steep retaining walls over most of the system here. These things go basically everywhere. You can kind of pick them out. If you just look on like this edge here around the spring island on the left-hand side, you can literally see the edge of that concrete in bright white along it. And that's that photo right there on the bottom. And so these are like 3-4 feet tall. If you're like a little musk turtle, I don't know, like, are you like rock climbing to get out of this? Like, and then you have to like hang upside down to get over the lip. Good luck. Down here at the lower end, at the end of Land Lake, is this structure here seen in this photo. It's in here on the aerial photography. This is that dam structure. It's steep. And basically, if you're a turtle, you basically go over it and then you have to like navigate your way back to the golf course or the residential area or the walking paths. And then you have to like free dive off of this 4-foot wall
So we
here.
got 5 minutes till the end of this.
We're almost there.
Session. Yep.
Yeah. And then we have some islands within the system as well, and those basically all got knocked out in the flood, so we're missing those as well. So basically, really depauperate on both access to terrestrial habitats that we need for turtles. We had some nesting habitat on these islands, those are gone as well. We're struggling. So if we look again at that big macro view here, and we can look at the land cover around it, we can start off in 1985 when this data set starts. We start with field forest in 1985 on our little graph down here, and this is just showing kind of the composition of land cover over time in that graph. What it says is we move from field to forest on the left-hand side, we basically move down into kind of fields and shrubs as we lose some of those forests, they're logged and things of that nature. We then basically get this period of fragmentation in the late 90s and early 2000s. And then basically after that, it's just this astronomical amount of developing, and you can just see that in the plot. Everything in gray is human development, and so we go from a very tiny little city, even up to the 1980s, lots of farms in yellow on the right-hand side of each one of these bubbles. You're gonna see that city expanding out. The brown in each one of these is called barren land from the National Land Cover Data Set. That's a giant cement plant. So that's not, it is barren, it's just being used by us. Oh, and just the main takeaway here, we go from 64% nature to less than 50% nature, 21% humans to 41% humans over that short period. So even within our time here, we lose a lot, basically a 9% gain in humans. Okay, so what's the gilded cage here? Let's bring it all the way back. We got tons of gilding, abundant food resources, invasive snails, everything's really nice for them. You can grow quick, you can get big, you can live for a long time. The faster you grow, the more likely you're able to evade predators. It's a spring, so it doesn't have a ton of like aquatic predators. There's not like alligators and stuff hitting them here. If you decide as an adult that maybe this luxurious oasis that I'm living in is not for me, maybe I want to go find someone else, or I can't find a mate because there's too many males out there, so I'm going to leave. You're basically not coming back because you're going over a dam if you can't even get out, because again, like 50% of the system's armored. If you can get out, there's not really much places for you to nest. If you can, basically the only place left is the north part of the golf course. So you're exposed on a golf range. If you can get there at all, that's pretty hard. Even for the place that's not armored, it is a karst system. So there's all this karst boulders around. So it already is limited under that. The whole entire landscape has changed, which is going to concentrate predators into this green space in the center of the city. And so we're pretty concerned about nest predation as a result. This can just whack out the entire year classes. And I think that's why we're seeing some variability in the number of turtles that are coming in, particularly the hatchlings. I suspect we're just not getting them anymore. 2017, where we see a lot of them, we kind of harped on them a little bit here. We lose a lot. It also happens to be the year that we lose those nesting islands. And so it seems like we lost islands and they were maybe the last bastion. And so just to put the finest point on it, good for growing, good for adults, super bad for reproduction. That's the gilded cage. We are not able to close this life cycle loop here for our turtles here. We're concerned about it. We're working on it. We're trying to figure out what predator densities actually look like here. What's the nest success if we can find them at all? And if we can't, then how do we build nest habitat for them? Which maybe we'll talk to some of you find environmental engineering folks. My lab does a ton of quantitative modeling. You've seen one example of that here. This is just a smattering of it. I work on sushi fisheries, so here's the sushi slide.
Yeah.
Thanks so much for your attention.
Thanks, Zach. That was great. Okay, we'll see if we have any questions on YouTube. In the meantime, questions from the room.
And I can go back to other slides if you want.
And Zoom also, we've got Lisa and Billy online if you have questions.
I have one quick question.
Yeah, please.
Were you only collecting the turtles from within the water and not around on the riparian buffer area?
Yeah, I
So,
mean, it would,
okay.
yeah, it would be,
Just to know.
that would be incredibly inefficient. Grabbing them in the water is 100% the way to go.
Also, if they couldn't escape
Yeah.
at first.
Yeah.
Place, probably
Yeah.
there.
Yeah, you don't see a ton of them here up on the shoreline. I mean, that's mostly all in water. Same with all the springs here. It's like
Mhm.
way easier to get them. If you're in other places, like if you're doing like big river systems, what they tend to do is they'll take a hoop net, which just has hoops and mesh between them, and they'll flip it upside down and weight the bottom of it and tie off the bottom and hang it underneath a basking spot. And then they'll leave and wait for the turtles to go bask, and then they'll swim up and swoop the turtles off into the net, and that's pretty efficient, because you
Yeah.
usually can't, if it's a really steep system, you can't keep the hoop net out of water. So the turtles end up drowning, which is defeats the point of mark-recapture. So you're into depletion sand.
Oh, well, you got a question here from, maybe you know this guy, Peter Frederick?
I know him.
Unless, it could be a different Peter
Yeah.
Frederick, but, what's being used on the golf course and might that affect breeding by turtles? So I assume he means some sort of, agriculture.
Yeah, they are using fertilizer. We're pretty concerned about some of the, I mean, in the residential area, we're concerned about just endocrine disrupting compounds in general. The fertilizer is definitely a problem. They keep this place exceptionally well manicured all the way up to the retaining wall, which is part of the, I think, one of the reasons they have it there, so they like run the mower across the edge. So it's not a great place to be putting turtles, so we would have to do like nest protection and do a bunch of things like that, mark those areas off. Which is pretty common for turtles. We'll see if the park jumps at that stuff. Edwards Aquifer Authority seems pretty concerned about what we're seeing, so there's some traction there, but they're not sitting around being like, oh no,
But it seems like it's, culturally and environmentally prioritized site, and so are there efforts to do habitat restoration?
There's a lot of habitat restoration happening there. It's kind of a classic system for like single species management and those trade-offs that tend to happen. I deal with this a lot on the high seas with protected species, where it's like, we want to save the whale, and whatever we do for the whale ends up causing us to catch sea turtles. And then we want to save the sea turtle and it hurts the birds, or don't fish here and then you fish there and you catch something else. So we see this happen a lot in single species management. In this case, fountain darters don't like the islands, so they're like, yay, the islands are gone. Great, more darter habitat. And it's like, well, there goes the turtles. Great. So there's some talks of doing some island restoration type process. They work really well when we bring them back. We do see that for turtles. Like if we can bring an island back, it basically can solve your problems. So it's a matter of getting that. There's some, much like our Florida Springs, they're kind of maybe like 10, 15 years, maybe even 20 years in terms of like where we were at on SAD. So they're still dealing with the problems of fertilizer and the problems of those things changing the spring systems. But they haven't quite moved as far down the trajectory as we have. So there's definitely a lot of work in the system. The little bryophyte mats are a pretty good indicator of like how bad it is or if it isn't, because they basically just get swamped with algae if the system's has a ton of nutrients in it. So, but it has a lot of flow. So at the moment, it's like
It looks
if
like
it has flow, it's doing good. It seems to bounce back from drought pretty well. So it still has the capacity. It's whether we can maximize that process here.
Other questions? All right, let's get it down.
If you do want to work on turtles, you can go to turtlesurvival.org, and they have under their About Us, they have a research page, and it says volunteer science. You can go there and you can volunteer and go and help out with snorkel surveys nearby. They sample manatee fanning, they sample Highland Blue Springs. It's an awesome experience. It's big fun. It's a pretty nice way to go and meet people also who are like doing science and want to go to grad school and stuff like that. So it's a good crew to get into, and there's a lot of people. And some of the folks who've been helping out with those surveys over years are on day games with us now.
Nice.
So yeah.
All right, thanks. That's good. Dr. Sanders, another round of applause. I appreciate it. All right, so we have 2 seminars left for the semester. I am miraculously basically caught up with your abstracts. Please do look at the abstracts for the comments, so like you do get a grade and, it's pass fail class or whatever, but I am giving specific feedback on your abstract, so next week we have Dr. Eni Hensel. And she's gonna talk about stingrays, so
Oh, awesome.
elasmobranchs. Elasmobranchs, so how stingrays and scars shape seagrasses.
Nice.
And so that will be in person here live in the room, so we will see you then, and thanks